The second thing, which is the structure. Now we gotta talk about the cells and the extracellular matrix. There's something special about the epithelial cells, which is called the cell polarity. Cell polarity. Which means, for example, if we have cells like that, given that these are epithelial cells, we would have different poles for those cells, and those poles will define different characteristics for each of those cells. So we have the apical pole, the one at the apex, and we have the basal pole, and there is the lateral pole. Sometimes the basal pole and the lateral pole are combined together, and the expression is called basolateral pole. So we have apical pole and we have the basolateral pole, or we can separate them like that. What does this cell polarity do? The cell polarity differentiates different structures inside the cell. So first of all, we have different proteins per se. So we have different proteins. In the apical pole, we would have channels, transporters, alright? The apical pole is the pole that's facing the lumen of whichever organ the epithelial tissue is covering from the inside. So, for example, if we have a blood vessel and the blood vessel is like that, this is a cross section, this part is the epithelial tissue, and the one that's facing the lumen, that part is the apical pole that we have in here zoomed in. So that apical pole usually would have proteins that would help the process of diffusion, to transfer things from in and out the cells inside the lumen or from the lumen to help the absorption as well. On the basal pole, on this part, which is facing the connective tissue of the vessel or the organ, we'd also have some channels of transporters, but the most important part, the specific part for the basal pole, is called adhesion proteins. So we'd have adhesion proteins that would help the cells to stick to the layers beneath them, which we'll talk about in a second. So this is for the proteins. For the lipids, as we all know that the membrane of the cells is made of phospholipids. So the lipids here are different. The distribution of the lipids in the membrane of the cell is different in the apical pole from the basal pole. The lipids in the apical pole, we would have cholesterol and sphingomyelin. So in the apical pole, we have cholesterol and sphingomyelin. At the basal pole, the lipids are phosphatidyl, choline, and phosphatidyl inositol, which also help the basal pole to be stable on the layers beneath. And we don't need that on the apical part because it's the free surface of the epithelial tissue. So that's for the proteins and for the lipids. We also have a difference in the distribution of the organelles inside the cells themselves. For example, the nucleus usually is resting more towards the basal pole. We have the protein synthesis organelles. For example, we have the rough endoplasmic reticulum, which is usually situated towards the basal pole. So here we usually have the rough endoplasmic reticulum. And then we have the Golgi apparatus, which is responsible for making the vesicles that would transport those proteins that were originally assembled by the rough endoplasmic reticulum. So the Golgi is usually surrounding the nucleus, and it's halfway through the cell and is closer to the apical pole. So this distribution is also affected by the cell polarity. And finally, we have the cell junctions. Now we all know that there are different types of cell junctions. Also, these are distributed according to the polarity of the cell. In the apical pole, we have tight junctions. So the cells from the apical part are usually closer together than in the lateral part. The lateral part is a bit more loose. So in the lateral part at the end we would have gap junctions, because there is a bit of a gap between those two cells here in the lateral pole. So let's start from up to down. First of all, in the apical pole we have tight junctions. Then in the lateral pole we would have adhesion built. We would have desmosomes as well. And the last one, which is the closest to the basal pole, so we can say that it's on the basolateral pole, we would have the gap junctions. The gap junctions. Then, on the basal pole, connecting the epithelial cells to the next layer that we're going to talk about, are the adhesion protein that we just mentioned here. We have two types of those. First of all, we have hemidesmosomes, and we have here focal adhesion. Those ones we call integrins. So this is the different distribution of the cell junctions, also according to the cell polarity.